this is Dr. Wu. Again, I will demonstrate the last lab, Lab 4, Mendelian genetics for segregation of monohybrid cross in corn. A monohybrid means that this cross is made only for study in one trait, meaning one gene. So this refers to what we learned this week about Mendelian genetic laws. The first one is the law of segregation. So it says two alleles will segregate during meiosis. We will use this law to study the trait in corn kernel. So let's look at the structure of corn kernel. So there are three layers here. The first one is pericarp. It is colorless. Then the second one is aileron. Aileron, the color of aileron can be purple or colorless. Then the endosperm is yellow or white. So it's controlled by a gene with two alleles. Let's say Y is dominant and little y is recessive. So when we see the color of different corn kernel, that due to the color of aileron. If this one is the colorless, then it will reflect the color of endosperm. If this one is purple, then we'll see the purple kernels. Another gene is the gene that control synthesis of starch. So in the corn kernel, the majority of them are this portion, that's the endosperm. So endosperm contains amino starch. So that's the main components. However, when this gene, sugarin gene, is in recessive mode, it will produce sugar instead of starch. In sugar, there are a lot of water. When the kernel is dried and the water evaporates, only left is the sugar, and the kernel will collapse because those volume of water has gone. And we'll see this wrinkled kernels. Then in this lab, we'll use a statistic test called the chi-square test to see whether or not collected data fit into the first Mendelian genetic law, which is a segregation law. So here is the formula for the chi-square test. So chi-square test is just for testing observed data to see whether it fits a certain ratio. So in Mendelian genetic law, the ratio of dominant versus recessive is 3:1. So we'll use this chi-square test to see whether the observed data for the trait in corn kernel fit first Mendelian genetic law. Here I need to explain the formula a little bit. O is for observed data, observed value, and then this E is expected value. So O minus E, that's the difference between observed value and the expected value. Then we square it and divide it by E again. And this symbol here, meaning summation, then we'll add all the O minus E squared divided by E together and get this chi-square. Then in order to do the chi-square, we need to get the degrees of freedom. So that's the symbol is TF. equals n minus 1. N is the category number. How many categories in this study? We have 2, then the degrees of freedom is 1. And for the statistical test, usually we'll set up a significant level, which is the probability. So in this case, we choose 5% or 0.05. Then this chi-square at this level is 3.85. So if they calculate the chi-square from the data smaller than this number, or p probability is greater than the significant level, then we'll accept hypothesis. Otherwise, chi-square greater than this chi-squared at 5%, then or the p smaller than alpha will reject hypothesis. Now, this is the introduction part for today's lab. Let's move on to see how we can collect the data and do chi-square test to see these two traits will follow segregation law. The first trait we study is the color of first trait we study is the color of the kernel. So we see those are the kernels. So I showed it, I remove it from a corn cap. There are just two colors. One is dark, so they produce the purple color outside, and another group is light. It's yellow color that we classify as light. So to study these two, to see whether the color of the kernel follow segregation law. So I group them according to their color. So there's two groups. I count the number of the kernels in each group. I record the number here. This is 128 dark kernels. Then this group, light kernels, there are 39 kernels in this group. Once we get this result, we can record this in the lab I provide on Blackboard. So this square here is Punnett square. It shows there are two gametes. So one from the parental father and another from mother, that's maternal gametes. When they come together through the fertilization, then we have 1/4 of dark, but the genotype is CC. That 1/2 is dark. The genotype is big C and little c. And the light color is little c, just 1/4. So I will fill this blank. So dark kernel is 3/4. Then the light kernels is 1/4. So this is a divide from 3 versus 1 ratio. The next one, I will fill out the table underneath. So this is observed number. Observed kernel is 128, and for dark group, then light group, that's and there are 39 kernels, and the total is 167. I calculate the expected number. So for the dark group, the expected number equals 167 times this portion, 3/4 and the result 125.3. And this number here is for the expected number in group, this light group. 167 times a quarter, the result is 41.8. The next one, I use O minus E divided by squared divided by E. That's this 128 minus 125.3 squared divided by this 125.3 again. So the result is 0.06. Then the same way we can get this chi-square for light group is 0.18. Add them together, that's 0.24. So this number is the chi-square number. It's a summation of chi-square, so that's 0.24. And the degrees of freedom, we have two groups, dark and light. So the degrees of freedom is 1. It's 1. 2 minus 1 equals 1. The next two numbers, this number here I got from the table attached, which is this one here, the last page. So we know the chi-square, the calculated chi-square in this table, calculated chi-square is on x-axis, so from 50 to 0. And the y-axis is probability that from 0 to 1. So in our case, the chi-square, calculated chi-square is 0.24. So it's here. Then I draw vertical line across the curve here with a circle 1. Circle 1 meaning this curve is for degrees of freedom of 1. So that's the correct one. Then I draw a horizontal line across y-axis here, then estimate the number. That's a 0.98. This way I got this number here, the estimate probability of fate by chance. That's P 0.98. Then the next question, is this P value smaller than P alpha? So this one is greater than alpha, which is 0.05. So the hypothesis for this test is this data fit 3 to 1 ratio. This result will make a conclusion that we accept the hypothesis because the p-value is greater than 0.05. So that's the process to get the data and finish the calculation for the first trait, the color of the kernel. Next one is the trait that relate to the shape of kernel. So this shape is controlled by the sugarin gene. If this one is dominant, then it will produce starch. Otherwise, when it's in recessive mode, it will produce sugar, and after water evaporate, the surface of the kernel changed, just collapse, form a lot of wrinkles. So you can see there's two type of kernel. This is full with a dominant allele, and this one with a recessive allele. So that's the wrinkled surface. The same for this colored kernel, and this is the normal one. Okay. According to this standard, I group the kernels from corn cap into this 2 group. So this is the sugaring, and the normal one is on the left side. Okay. I counted the number of kernels, 125 in normal group, and this 36 kernels in sugaring group. And we repeated the same procedure. So I write these 2 ratios, because the sugaring and one from this parental parent, another from paternal parent and maternal parent. Then this the offspring has a different genotype. So sugar either in this homozygous dominance or heterozygous, or 2 of them. So add them together, that's a 3/4. Then 1/4 is for the sugaring. And I write this observed number here, 125 and 36, and this total number is 161. The expected number are those 2. The assignment for the students finish calculating the chi-square, use this formula, and get the total here, and write it down on the first blank, then the degrees of freedom, and use the attached table to find the estimate probability. Then compare this one with 0.05 to see it is greater or smaller than 0.05, and the hypothesis is the same. Shape of the kernel tested here fits 3:1 ratio. Do you accept or reject it, and why? So that's all for lab 4. If you have any question, please email me. I hope you're enjoying the labs. That's all for today. Bye bye.